Well, good morning. I'm Ian Walmsley, the Provost at Imperial College London and a sometime Professor of Experimental Physics, and I have the pleasure of chairing the science session this morning. This is a really fantastic opportunity for new science and for the research base in the UK, and because in this country a lot of the research base is within universities, it's a really important opportunity for the university sector. It's also appropriate that we're holding this meeting here because it is the basis of these new tools that enable new discovery. And in turn, it is the new science that gives rise to new technologies, and always in places and in areas that are utterly unexpected. So it will be a very important step to have this new tool to take us beyond where we can currently probe with light. So we will move to the session which starts with 7 talks covering areas of science that the XFEL could help with. And those range from the subatomic to the galactic. So hold on to your seats and prepare to be wowed. 15 minutes each, 7 speakers without hesitation, deviation, or repetition. So let me call upon the first talk from Alan Orville from Diamond Light Source, who will give us an insight into life sciences. Alan.
Yeah, so thank you very much. He just told me he'll wave at me at 45 minutes. Is that right? No. So thank you very much. I'm Alan Orville. I am the XFEL Hub group leader at Diamond Light Source, and I represent life sciences. Should I use a pointer? Yeah, so I don't work alone. I had help from Jasper and from Xiaodong and a lot of contributions from the science community. And so I think it's important to say thank you to the community for their contributions and for their activities in this regard. So when you think about structural biology, almost always you think about atomic coordinates. And those coordinates come from usually a crystalline sample held at 100 K. And it's been tremendously successful over the years. 200,000 structures have been deposited in the protein data bank. And there's a growing number of cryo-electron microscopy structures as well. And free electron lasers are the new kid on the block with about 600 structures or so. And there's an unknown number of structures that are in the pharma vaults. So there's a lot that's in the public domain, but there's also a lot that's in the private domain. So 100 K is a little bit like the surface of Pluto, or really the dark side of the moon. And so what really for us is the remaining frontier challenges in structural biology is to determine time-resolved atomic structures of systems engaged in catalysis, perhaps even in the native context. That would be complex and interesting, or their environment, and certainly at physiological temperature and pressure on Earth. So we want to be room temperature, one bar, although there's some interest in the organisms that live on the surface of whales that go so deep diving. And what does the biology take to go from the deep depths of the ocean up to atmospheric conditions and not denature? So there's a lot of really interesting biology out there. And so I like this quote also from Richard Feynman: all things that are made of atoms, and it's that wiggling and jiggling that makes the biggest difference for those molecules. And it doesn't ever escape me that crystallography started in the UK. I happened to meet Max Perutz when I first came to England, but John Kendrew and those folks discovered or determined the crystal structure of myoglobin in 1961. It took them years, and this is how they presented that result in Scientific American. Somebody physically had to paint a model of what somebody else had built in 3 dimensions. This is before crystals. This is what we do now. This is from Ilma Schlichting and was published in 2015. So this is a crystal structure of a time-resolved serial crystallographic approach where they hit it with laser light just before the X-ray pulse came in. And look at this. Instead of 1 or a few crystals, it took 96,500 individual lattices merged together to give the one ground state structure, and then 20,000 lattices for each of the time points. And they can see the dissociation of an oxygen-like mimic, carbon monoxide, after that laser pulse. And so free electron lasers, this is from Abbas, free electron lasers enable 3 major transformations: from overwhelming data to useful information, from a static structure to dynamic objects, and from gibbling objects to purposeful function. And I think that's nicely illustrated by this. And this is where we can go and continue to go. And that's where we're going to go. So the protein database, I don't want you to read this whole slide, but I want you to realize that it's a rich history. It's been around for about 50 years. And on the left-hand side are things that are related to the PDB, and on the right-hand side are some of the landmarks that came through there. And there's probably 12 or 13 Nobel Prizes that are in this list. And so we get to 2021 and you say, all right, that's 50 years of the protein data bank. Are we done? It turns out that this is an amazing investment, roughly $20 billion in this infrastructure for the world, in the public domain. And at almost more than 95% of all FDA-approved drugs now use molecular structures at their heart. But 2021 was also really important too, because that's when AlphaFold 2 was released. And so this is where the artificial intelligence comes into and developed by DeepMind and published in Nature. They're predicting the 3-dimensional structures of proteins based on the amino acid content. And so they say this problem is solved, in quotes. And it is very, very good for monomers and resting states, but it doesn't really do anything with ligands or cofactors. So it is a really important release. And sorry, I meant to say that they've also released the code and 200 million predicted structures with some estimate for confidence and uncertainty in those. So this is a game changer. This problem isn't solved. AlphaFold 2 and Rosetta Fold are very good at hypothesis generation. So it is having an impact and it will make an impact, but we still have a lot of changes remaining. Dynamics, function, reactions, drug discovery, the interactome. If it took 200,000 structures to get to the point of AlphaFold for resting state structures, how many structures do we need for time resolved studies to get to the dynamics? And that's what free electron lasers can bring us. The opportunity to study function and structure from the same sample and the same X-ray pulse. So the era of dynamic structural biology, that's where I say we are now. And it really is going to be time resolved structural biology. There'll be high rep rates and there'll be some impact across the board for this case. And I don't think we're done. There's 2.2 billion WGSs, which is shotgun approach sequences for sequencing the metagenome around the world. So there's many, many, many amino acid sequences that are still to be explored. 6 million enzymes, more than 30 million genes. And time-resolved structural biology is generalizable. We're interested in ground state structures, but also how those structures change as a function of reactivity. And we're going to follow this with multiple kinds of techniques. I mean, the crystallography is great. You can see where all the individual atoms are, but what's the chemistry happening at the iron or the oxygen? And so it's following the spectroscopic signature. We can get information on the electronics and the atomic structure from the same sample and the same X-ray pulse. That's really, really game changer. Another good science case is the oxygen evolving complex in photosystem 2. My collaborators and I, mostly my collaborators, are looking at the changes in the redox state of this complex iron manganese cluster as it gets oxidized by photons. And eventually, these two atoms right there, number 04 and 05 and 06, come together to make molecular oxygen. That's what we're breathing right now. This chemistry changed the planet as the oxygen evolving complex evolved in organisms. So what about deeper insights into biology and pharma and clean energy? And here I'm going to focus in particular on membrane proteins. And I'll mention G-coupled protein receptors, which is a wide range of families, that 800 human different proteins and thousands of different ligands that impact every aspect of human health. But the reality is it's great to have resting state structures. Where do we need to go? Dynamics. And so how do you do that? With some kind of a mixing injector that will allow us to bring together those membrane-bound proteins and add ligands and measure the dynamics, the changes of the structure that will give rise to how you may monitor your blood pressure, for example. So then we can also think about water-soluble proteins and how antibiotics bind into enzymes that degrade those antibiotics. So the human health impact is huge in this particular area. If we don't continue our antimicrobial research, some predictions are 10 million deaths per year in about 2050. So this is a huge area of research, something that we're quite interested in. And you might notice in this slide, we can do some studies at 2 milliseconds of time resolution, which is right in the sweet spot for biology. We also do complementary results at diamond light source at 12 milliseconds. So my theme here is, what is one of the impacts of free electron lasers? How it changed we do everything at synchrotrons. It really is important how the technology transfer between the different facilities is really key to science. And so you'll hear from me that we're trying to duplicate the free electron laser experiment at Diamond so that we can train our user base and do appropriate experiments and then complement the free electron lasers. So this is a common theme for us. So this is me, and I was brought in 2015 with soft money from the Wellcome and the MRC and BBSRC, and a contributing fund from Diamond Light Source. And over the years, I've been able to make connections to different research groups around the UK. And now from 2019 and forward, we've got most of the funding from the Wellcome Investigator Award and the Royal Society, and some from the UKR and BBSRC, and Diamond is also kicking in. That's allowed us to grow the group. This is a picture from 2022 last summer, and I was really happy. You notice how everybody's smiling? Even though these are stressful experiments, they're fun to do, and it really makes a difference for the young people. These guys are why we're thinking about a free electron laser, not me. It's for the next generation of scientists. And so this is the funding profile, and these are the tasks that we do, and we're roughly even split, but we're saturated. We can't do a lot more. We need a lot more effort to grow this community effort. Nevertheless, I can tell you that we have had some impact in the UK community. When I first showed up in 2015, it was 3. It was myself, and I went on 3 experiments. But luckily, this has grown over the years that now we have a really good number of scientists that actually go and participate in free electron laser research. And moreover, many of these collaborators are now winning funding from other agencies to enable their science and technology. There's several even in the room I can point to and say, yep, you got money, you're working in this area, and we're working with you. And so another way to think about impact is a baker's dozen of papers. And those are papers that came out with some fingerprint of the XFEL hub, and they have some reasonable profile to them. And so this is the impact that we're doing through the hub. And to that, I need to say thank you to the funding agencies. So the wish list for science for the machine, there's going to be another slide that gets into the end station. These slides will be available to everybody, and I don't want you to get into the details here, but I want you to understand that there are some things we can do now, and there are some things we need to do in the future. And a shorter pulse duration under a femtosecond could be quite helpful and allow us to get into attosecond time resolution, especially for ultra-fast chemistry. That can't be done very well. And that impacts some really important light-activated systems in biology. We do need a reasonably good repetition rate, but I would also say that a lot of this is limited by the detector. Why do you need a megahertz machine if your detectors see in the tens of kilohertz? Well, if you can share the beam, that's one way to do this. We do want to work at high energy, and we want a variety of different pulsing sequences, including even chirped energy pulses that give us much pinker beam, more like a Lowry time-resolved experiment. We really need good energy because in some cases, if you've got crystals, that's an amplifier effect. But as you shrink those crystals, your Bragg spots get weaker. So we do need some good flux density. But we also need good flux density if we're doing complementary experiments. X-ray emission spectroscopy tends to be a little bit photon starved. And so we want 2-color mode, and we need a really small focused beam. So if you then think about, so I think I'm almost done. If you think about the end station components, we have a whole range of things, and you can't forget those end station components. They're important for how you do it. And perhaps one of the bigger ones is how do we have enough high performance computing methodologies available so that we can process data and get some near to real-time feedback for our results in the trenches. So there's a lot of things that we need from the end station components. And finally, I want to finish with just a little bit about what we're doing at the hub to illustrate the similarities between diamond and free electron lasers. So we have two projects. One of them is this 713, and it's a sample delivery project where on demand, we put droplets onto a conveyor belt, we bring those droplets into the beam, and in the forward direction, we collect X-ray diffraction data, and at 90 degrees, we collect, there it is, X-ray emission spectroscopy. So this is going on at Diamond right now. But what is this really? It's a prototype end station for training and evaluation to be sure that it all works before we roll it out and try to do user opportunities at XFEL. So the synergy between the synchrotron and the free electron laser is really, really strong. And you'll make better use of a free electron laser if you've got your instrumentation really worked out well at a synchrotron. And that's one of the things that we're doing at Diamond. And so this is my last slide to give you a little sense of where some of the things we're considering. It's not set in stone. I edited it a few minutes ago. And I need feedback from everybody, right? We need feedback from everyone. So we need good, stable, long-term funding from the hubs. I'll say that in plural, so that we can train PhD students, postdocs, and the user community. That's clear. We really need that. We're trying to take this end station component and take it out of the yellow school bus that it's in now and put it in a more permanent hutch, but it's unfunded. So we need ways to fund this so that we can make a better home for serial crystallography and soft condensed matter at Diamond. We are actively working on a collaboration with XFEL so that our instrumentation will be moved and used in this Crystallina end station in the short term to enable 100 Hertz data collection for correlated time-resolved crystallography and X-ray emission spectroscopy. And the European XFEL, they've got some unfinished tunnels. There's some things we can do there, and it's up for us in the community to decide what's that. So for that, I'll stop and say thank you very much.